We are looking at Chapter 27, which is a definitive text on chlamydia species. And honestly, calling this thing just an STI is like calling I don't know, a nuclear submarine just a boat. I mean, it technically fits the description, but it misses all the insane engineering that makes it special.
That is a very apt comparison, because when you actually pop the hood and look at the biology of chlamydia, how it lives, how it eats, how it hides, it is an absolute biological anomaly. It breaks the rules. It really blurs the lines between what we think a bacteria is and what a virus is.
The text actually opens with a phrase that completely stopped me in my tracks. It defines chlamydia as an obligate intracellular pathogen. Now, that sounds like a lot of dense medical jargon, but it's actually the key to everything this bug does, isn't it?
It is the foundational definition, yeah. Let's break it down a bit. Most bacteria that people are familiar with, think E. coli, Staph aureus, Salmonella, those are what we call free-living organisms. They are independent contractors, essentially.
They're totally self-sufficient.
Right. If you put E. coli in a petri dish with some sugar and water, it's perfectly happy. It eats, it grows, it divides. It can live on a doorknob. It can live in a sandwich. It can live in your gut. It has its own survival kit. Chlamydia does not. The word obligate means it has absolutely no choice. It is obligated to live inside a host cell.
So if I just put chlamydia on a table, it dies.
Well, it creates a survival form, which we will definitely get into, but it cannot grow. It cannot replicate out there. It is strictly confined to the interior of a eukaryotic cell. It's not just visiting. It literally requires the host machinery to exist.
Which brings us to the second label the text uses, which is even more evocative, and that is energy parasite.
Yes. This is the crux of why it's an obligate pathogen. To understand this, we have to go back to high school biology for just a second. Do you remember ATP?
Adenosine triphosphate, the powerhouse of the cell, right?
Exactly. The mitochondria making the powerhouse currency. ATP is the universal energy currency of life. Every chemical reaction in your body, from flexing a muscle to firing a neuron, requires you to spend ATP. And bacteria are exactly the same. They need ATP to build their cell walls, to replicate their DNA, to do literally anything.
And usually bacteria just make their own.
Usually, yes. They have all the metabolic pathways, things like glycolysis, the Krebs cycle, the electron transport chain to generate this currency from raw materials. Chlamydia does not have those.
It's just broke.
It has absolutely no currency of its own. It lacks the fundamental enzymes to synthesize ATP. So it has to steal it.
It's a thief.
It is a literal energy parasite.
Right.
It relies entirely on the host's sure cell to furnish energy-rich intermediates. It effectively plugs itself into your power grids, like a squatter who moves into your house, not only sleeping on your couch, but running an extension cord to your generator because they didn't bring any batteries of their own.
That seems, well, it seems incredibly lazy, but also incredibly risky. Because if the host dies or runs out of energy, the bacteria is completely done for.
It is risky, but it's also highly, highly efficient. We call this reductive evolution in biology. Think about the massive cost of maintaining a power plant.
Yeah.
It takes up physical space, it requires resources, it requires constant maintenance. By just ditching all that metabolic machinery, chlamydia can travel light, it can be small, sleek, and focused entirely on one thing, which is replication.
So the mission of this deep dive is to really understand this energy parasite. And the scope of the file we're looking at is way broader than I thought, too, because we aren't just talking about one bug.
No, we are looking at the genus Chlamydia as a whole. The source material highlights 3 specific species that are pathogenic to humans.
Right. We have Chlamydia trachomatis, which is the famous one causing the STIs and, as I learned, severe eye infections. Then we have Chlamydia pneumonia,
which, as the name clearly implies, targets the respiratory system, the lungs.
And the third one, which I frankly did not know existed until today, is Chlamydia spp.
The zoonotic threat.
Zoonotic, meaning it jumps from animals?
Specifically birds. That's the agent of parrot fever.
Parrot fever. We are definitely going to talk about the parrots later. I desperately need to know how one gets parrot fever. But before we get to the specific diseases, I want to stay on this alien biology track. Because the first section of our outline here is all about the structure and the metabolism, and there's a quick note here about taxonomy that I want to clear up.
Ah, yes, the taxonomic wars.
Yeah, the text mentions a controversy about splitting the family into Chlamydia and Chlamydophila.
Taxonomy is always in flux as we get better genetic sequencing tools. For a while, there was a really strong push in the scientific community to separate pneumonia and Cetacea into a brand new genus called Chlamydophila. You will still see that name pop up in a lot of research papers from the early 2000s.
But our source text says let's not do that.
The source text acknowledges the debate. but it notes that the split hasn't been universally accepted, and honestly, it can be really confusing for clinicians on the ground. So, for the sake of clarity and our own sanity today, we are going to follow the chapter's lead and refer to all of them under the single genus Chlamydia.
Good. Keep it simple. Now let's talk about the gram-negative paradox, because this was another one of those wait, what moments for me.
It's a classic microbiology riddle, really.
So usually we divide bacteria into two big camps, right? Gram-positive and gram-negative, and that's based entirely on their cell wall.
Correct. It is the very first thing you learn in medical school microbiology. Gram-positive bugs have a very thick, mesh-like wall made of a substance called peptidoglycan. Gram-negative bugs have a much thinner wall, but they compensate by having a second outer lipid membrane.
So where exactly does chlamydia fit in that?
Well, if you look at it under an electron microscope, it looks gram-negative. It has that distinct double membrane structure. It has a high lipid content. It even has lipopolysaccharide, or LPS, embedded in that outer membrane.
And LPS is the absolute hallmark of gram-negative bacteria. So, case closed, right?
You would think so. But the defining structural feature of a bacterium is that peptidoglycan wall, that mesh. It's what gives the cell its physical rigidity. It protects it from osmotic pressure so it doesn't just absorb water and burst. Chlamydia, incredibly, appears to completely lack this wall.
It's just naked.
In the space where the wall should be, between those two membranes, we don't find the typical peptidoglycan structure. For a really long time, scientists thought it had none at all. But here is the paradox.
The penicillin connection.
Exactly. Penicillin is an antibiotic that works specifically by targeting the enzymes that build the peptidoglycan cell wall. These enzymes are literally called penicillin-binding proteins. If a bacteria has no wall to build, it shouldn't need those proteins, and penicillin shouldn't do anything to it.
But the text says penicillin actually does inhibit them.
It does. If you treat a chlamydia culture with penicillin, it stops dividing properly. It doesn't kill them efficiently, which is why we do not use penicillin for treatment, but it creates these massive, bloated, aberrant, defective forms.
So the bacteria has the tools to build a wall, and it reacts to the drug that stops the wall, but we literally cannot find the wall.
Right. It strongly suggests that chlamydia maintains a very minimal, perhaps highly transient, peptidoglycan structure. Just enough to help it physically divide during replication, but not enough to be detected by standard chemical means. It's almost like a ghost wall. It's there exactly when it needs to be and gone when it doesn't.
That is bizarre. It's like it's wearing the heavy coat of a gram-negative bacteria, but wearing absolutely nothing underneath.
That's a great way to picture it. And speaking of that coat, we have to mention MOMP.
MOMP. It sounds like a cartoon sound effect.
It stands for Major Outer Membrane Protein. This is the absolutely dominant protein sitting on the surface of the bacteria. And it is incredibly crucial for us as scientists and doctors because it serves as the primary ID badge.
How does that work?
MOMP is highly variable. Its specific structure changes slightly from strain to strain. In Chlamydia trachomatis, we actually use the molecular structure of the MOMP to classify the bacteria into what we call serovars.
Serovars? That's kind of like a subspecies.
Think of it like a sports team. The overall franchise is Chlamydia trachomatis, but the individual players wear different jersey numbers. The jersey number, the serovar, tells you exactly what position that specific bacteria plays. Does it attack the eye? Does it attack the reproductive tract? Does it attack the lymph nodes? The MOP tells us the answer to that.
So genetics is entirely destiny here. And speaking of genetics, you mentioned earlier that it travels really light.
Extremely light. It possesses one of the smallest genomes of any free-living or pathogenic bacteria known.
Give me the actual numbers here so we can grasp the scale.
The genome is roughly 1.04 megabases. That translates to coding for only about 900 individual genes.
900 genes? That seems incredibly small for something that does so much damage.
It's tiny. For comparison, a standard E. coli has over 4,000 genes. Humans have somewhere over 20,000. Chlamydia has ruthlessly stripped away everything that isn't absolutely essential for its specific parasitic lifestyle. It threw away the genes for synthesizing amino acids. It threw away the genes for making ATP. It made an evolutionary decision that it was much more efficient to steal than to create.
But it did keep some genes for weapons, because the text highlights something called the type 3 secretion system.
Yes, this is a critical piece of machinery. Despite being so genomically small, chlamydia is heavily armed. The type 3 secretion system is essentially a molecular syringe. It's a microscopic needle complex that projects out from the surface of the bacteria.
And what does it do with this microscopic needle?
When the bacteria comes into physical contact with a host cell, It uses this system to literally puncture the host's cell membrane. But it doesn't just poke a hole. It injects specific effector proteins directly into the host cytoplasm.
Like injecting venom.
It's vastly more sophisticated than venom. Venom is generally just designed to kill or paralyze. These proteins are command signals. They hijack the host cell's internal scaffolding, the cytoskeleton. They force the host cell to rearrange its own structure and actively reach out and engulf the bacteria.
That is absolutely terrifying. It's not just knocking and asking to come in. It's grabbing the steering wheel and driving the car right into the garage itself.
That is a perfect metaphor. And that garage concept leads us directly into the life cycle, because the second section of our outline is entirely about this, and honestly, this part reads like a sci-fi horror script.
I thought the exact same thing when I was reading it. The text calls it the biphasic developmental cycle. 2 phases. 2 completely different phases.
It's totally unique. Very few bacteria have a life cycle this complex and dynamic. It takes about 48 to 72 hours to complete one full round. You have 2 distinct morphological forms, the elementary body, or EB, and the reticular body, or RB.
Okay, let's visualize this for everyone listening. Step 1, the elementary body, the EB. This is the one you actually catch, right?
Correct. The EB is the infectious form. It's the traveler. It's very, very small, about 0.3 micrometers, and if you look at it under an electron microscope, it looks incredibly dark and dense.
The text mentions disulfide bonds here again.
Yes, remember we said it essentially lacks a functional cell wall. Well, to survive outside the human body, the EB highly cross-links its outer membrane proteins with disulfide bonds. Think of this as the bacteria putting on a rigid suit of chainmail armor.
So it's heavily armored for the outside world.
It is incredibly tough. It's highly resistant to environmental stress.
Yeah.
It can survive the cold, the dry air, the physical journey from one person to another. But there is a massive biological trade-off. In this heavily armored state, it is metabolically completely inert.
It's just a rock.
It's a spore-like rock. It doesn't eat, it doesn't divide, it just hunts. Its single, solitary purpose is to find a susceptible host epithelial cell and attach to it.
Okay, so the EB hits the cell, uses that molecular needle we just talked about, injects the proteins and gets sucked in. Now it's inside the cell. It's inside a little membrane bubble called a vacuole.
Or a phagosome, yes. Now, this is the absolute moment of truth for the pathogen. Normally, when a human cell sucks in a foreign bacteria, its internal defense mechanism kicks in immediately. It tries to fuse that bubble with a structure called a lysosome.
What exactly is a lysosome?
A lysosome is basically a cellular bag of strong acid and digestive enzymes. It's the stomach of the cell. If the lysosome fuses with the vacuole holding the bacteria is instantly dissolved and destroyed. It's game over.
But chlamydia survives this.
Chlamydia cheats the system. The EB, once it's safely inside the vacuole, sends out specific chemical signals that actively prevent that lysosomal fusion. It blocks the stomach from attaching. It turns that vacuole from a death chamber into a highly protected safe house.
That is incredible. So it's sitting in the bunker, it's safe from the cell's defenses. What happens next?
Phase 2. The transformation. The disulfide bonds that heavy chainmail armor physically relax. The bacteria begins to swell up, gets much bigger, turning into the reticulate body, or RB.
And the RB is the factory.
Exactly. The RB is the replicative form. It is highly metabolically active. This is the stage where it actually plugs into the host's ATP supply. It starts eating, and it starts dividing by binary fission.
Oh, it just clones itself over and over.
Rapidly. But here's the critical vulnerability. The RB has no armor at all. It is incredibly fragile. If you took an RB out of the host cell, it would pop and die almost instantly.
Yeah.
It can only exist inside that carefully controlled safe house.
So the RBs just keep dividing and dividing. They fill up the bag?
They fill it until the vacuole is absolutely massive. In microbiology, we call this an inclusion. If you look at a slide of a heavily infected cell, you literally see this huge, bulging bag of bacteria pushing the host's own nucleus off to the side. It takes over almost the entire internal volume of the cell.
The cell is just completely stuffed full of these things. And then what? It can't stay in there forever.
The exit strategy. The bacteria somehow know likely, through sensing that the host's ATP or nutrient resources are running critically low, that it's time to leave. So the fragile RBs start to condense again. They transform back into EBs. They put the armor back on.
They suit up for the outside world.
They suit up. And then the host cell lyses.
Lysis meaning it bursts open.
It forcefully ruptures. The host cell dies, ripping open to release hundreds or sometimes thousands of newly minted, fully armored EBs into the surrounding tissue.
Like a biological grenade going off.
Exactly. And each one of those newly released EBs floats away to infect the neighboring healthy cells. And the entire 72-hour cycle starts all over again.
It is terrifyingly efficient. A relentless loop of invasion, theft, replication, and total destruction.
It really is a marvel of evolutionary design, as destructive as it is.
Okay, so that is the machinery. That's the how. Now I really want to get into the what. The actual diseases this causes, because Section 3 of our outline focuses specifically on Chlamydia trachomatis, and this is where those jersey numbers, the serovars, really come into play.
It is fascinating how a single bacterial species can cause such vastly different diseases just based on slight genetic variations in that surface protein. We generally categorize C. trachomatis into 3 broad clinical groups.
Let's start with the very first group, serovars A, B, B, and C. The text labels this group trachoma.
Trachoma. This is a disease with an incredibly deep human history. The source material notes it was accurately described in the Ebers Papyrus in ancient Egypt, that is 3,800 years ago.
Humanity has literally been dealing with this specific bug for nearly 4 millennia.
And tragically, we still are. Trachoma remains the absolute leading cause of infectious blindness in the world today. It's hyperendemic in areas with poor sanitation and infrastructure, parts of sub-Saharan Africa, the Middle East, parts of Asia. It completely thrives in environments where clean water is scarce.
How does an eye infection spread so effectively in those environments?
Mostly through direct contact, finger-to-eye contact, sharing unwashed face towels. And a major vector is flies. There are specific species of flies that are attracted to and land on the faces of children, feeding on the purulent discharge from their infected eyes, and they physically carry the armored EBs from child to child.
That is a deeply disturbing image. Now, regarding the actual mechanism of the blindness, I always just assumed the bacteria itself ate the eye tissue, but reading the text, it's vastly more complex and tragic than that.
It is. The bacteria itself only infects the conjunctiva, which is the thin mucosal lining of the inner eyelid. But the permanent blindness isn't caused by the bacteria eating the cornea directly. It's caused entirely by the host's own immune system.
The immune system again.
The persistent infection causes chronic, repeated inflammation. We are talking years and years of constant low-grade inflammation, and a fundamental rule of human biology is that when tissue is inflamed for that long, it eventually scars.
And scar tissue is rigid, it's tight.
It contracts heavily. Imagine a bad burn scar on your skin shrinking as it heals. Now imagine that exact process happening on the delicate inside of your eyelid. As that massive scar tissue tightens, it physically curls the entire margin of the eyelid inward.
This is the medical condition called trichiasis.
Yes, the eyelid rolls in. So now, your eyelashes, which evolved specifically to protect your delicate eye, are turned completely against you. Every single time you blink, those stiff lashes sweep across the highly sensitive cornea like a wire brush.
Oh, that makes me physically cringe just thinking about it. That sounds agonizing.
It is incredibly painful. Constant, unending pain. And over time, that ceaseless mechanical abrasion completely scratches and destroys the clear cornea. It clouds over entirely. We call it corneal opacity. And the person goes permanently, irreversibly blind.
So the bacteria just starts the fire, but the resulting scar tissue does the actual catastrophic damage.
Precisely. The text cites some staggering numbers. Roughly 400 million people actively affected and 20 million completely blinded. It is a profound tragedy of poverty and lack of hygiene infrastructure.
That's serovars A through C. Now, let's move to the group that most listeners in the developed world are intimately familiar with. Serovars D through K. The genital infection.
The silent epidemic. These are the specific serovars that have adapted perfectly to the human reproductive tract.
In men, the text says it primarily causes non-gonococcal urethritis, or NGU, and epididymitis.
Right. It presents with painful urination, a purulent discharge, localized swelling. It's highly unpleasant. But, and this is a crucial distinction for public health because it is so physically painful, men usually notice it quickly. They go to the local clinic, they get tested, they get treated.
But in women?
In women, the clinical story is often completely different. It infects the cervix and the urethra. But the source material emphasizes one very specific vital phrase. Subclinical infection is the rule.
Subclinical meaning absolutely no symptoms.
Completely asymptomatic. A woman can be fully infected. She could be actively carrying a high bacterial load, shedding it to her sexual partners, and have absolutely no idea. She feels perfectly fine.
And because she feels fine, she doesn't go to the doctor to get screened.
Exactly. And that is exactly where the severe long-term danger lies. Because silent, definitely does not mean harmless. The bacteria is still very much there, actively replicating, bursting cells, and triggering that same low-grade chronic inflammation. And if left completely untreated, that bacteria can eventually ascend.
It physically moves up the reproductive tract.
It travels up from the cervix into the highly delicate uterus and the fallopian tubes. This causes pelvic inflammatory disease, or PID.
And just like we saw in the eye with trachoma.
Chronic inflammation leads directly to scarring. But here, the devastating scarring is occurring deep inside the fallopian tubes.
The incredibly delicate tubes that carry the egg from the ovary to the uterus.
Those tubes are microscopically tiny. If rigid scar tissue forms in there, it can block them completely. That leads directly to permanent sterility. The sperm can simply never physically reach the egg.
Or, arguably even worse, what if it's only a partial blockage?
Then you risk an ectopic pregnancy. This is an absolute nightmare scenario clinically. The tiny sperm can sometimes swim up and get through the partial blockage, and it successfully fertilizes the egg. But the fertilized egg quickly becomes much larger, and it is entirely too big to get back down past that rigid scar tissue. So it implants right there inside the thin tube.
And the fallopian tube cannot stretch and grow like the uterus can.
No, it cannot. As the embryo grows, it inevitably causes the tube to rupture. This leads to massive, sudden internal bleeding. It is a catastrophic, life-threatening medical emergency that requires immediate surgery. And it all stems from an infection that started out completely silent.
It's just devastating. And the text also mentions that it affects the next generation, too. Neonatal transmission.
Yes. If a pregnant woman has an active, untreated infection in her cervix, the baby is highly exposed as it passes through the birth canal during delivery. Roughly 50% of infants born to infected mothers will acquire the infection in some form.
What exactly happens to the newborn?
The first presentation is usually 5–12 days after birth. You see what's called inclusion conjunctivitis. The infant's eyes get heavily swollen and have a thick, sticky, mucopurulent discharge.
Sticky eyes.
Very sticky. But the much more serious potential complication comes a bit later. 2 to 12 weeks out, the infant can develop a specific type of infant pneumonia.
Pneumonia in a newborn is incredibly scary.
It is, and the text describes a very distinct clinical presentation that pediatricians look for, a staccato cough.
Staccato, like short, sharp machine gun bursts.
Exactly. Very short, repetitive, exhausting coughs. But curiously, the baby is almost always afebrile, meaning they have no fever at all, which is unusual for pneumonia. And if you look at their blood work, you see a massive spike in eosinophilia, or a rise in a very specific type of white blood cell. If a doctor sees an infant with a staccato cough, no fever, and sky-high eosinophils, they should immediately think chlamydia.
Okay, so that covers serovars D through K. We have one more distinct group within C. trachomatis, serovars L1, L2, and L3. This is lymphogranuloma venereum, or LGV.
LGV is the truly nasty cousin. The other strains we just talked about, trachoma and the standard genital strains, they are primarily mucosal. They prefer to stay right on the surface lining of the tissue. LGV is highly invasive.
Invasive in what way?
It does not just sit on the epithelial skin, it actively penetrates. It dives deep into the underlying tissue and aggressively targets the lymphatic system.
The lymph nodes.
Yes. It usually starts with a very small, painless ulcer at the site of infection that the person might not even notice. It heals up very quickly. But the bacteria hasn't left. It has moved on. It travels quickly through the lymphatic channels to the regional lymph nodes, most commonly the inguinal nodes in the groin.
And what happens when it gets there?
The lymph nodes swell massively. They become tightly matted together and are incredibly painful to the touch. They form what are historically called buboes.
Buboes. That is a word I mostly associate with medieval history and the bubonic plague.
It's the exact same physiological concept. These are massive, swollen, pus-filled lymph nodes. And if they are left untreated, they can actually rupture spontaneously. They burst right through the overlying skin, creating massive draining sinuses. It causes severe suppuration, heavy pus formation.
That is a wildly vivid and horrific image.
It gets significantly worse. The profound scarring from this massive deep tissue inflammation can completely, permanently block the lymphatic drainage of the entire pelvic region.
And if the lymph fluid physically can't drain back up the body,
you get profound swelling. In severe chronic untreated cases, this can literally lead to massive elephantiasis of the genitals. Or, if the primary infection was located in the rectum, which is increasingly common in women or men who have sex with men, it can cause severe rectal strictures and complex fistulas. It is a highly destructive, deeply invasive disease.
It is frankly amazing that this is technically the exact same bacterial species as the one that causes a completely silent, unfelt infection in the cervix.
It really highlights the sheer power of those tiny virulence factors.
Yeah.
Just a slight evolutionary tweak in the MOMP protein, a few internal genetics, and you go from a perfectly silent passenger to a highly invasive, tissue-destroying monster.
Okay, let's widen the lens a bit. That was all Chlamydia trachomatis, but we have two entirely different species outlined in the source material. Section 4 covers Chlamydia pneumonia.
The primary respiratory agent.
How is this one structurally or biologically different from what we just discussed?
Well, for the microbiology lab geeks out there are two really key differences. First, visually under the microscope, the elementary body is distinctly described as pear-shaped.
Pear-shaped, so it's not perfectly round like the Trachomatis EBs.
Right. And second, the large intracellular inclusions. Those massive bags of replicating bacteria inside the host cell do not contain any glycogen.
Why does the presence of glycogen matter at all?
Because one of the really old-school, very cheap ways to find a chlamydia infection under a microscope was to flood the sample with an iodine stain. Iodine binds to glycogen and turns it a very dark, easily visible brown. So C. trichomatis inclusions stain dark brown. C. pneumonia inclusions do not. They stay completely clear.
So it's effectively a negative visual test.
Exactly. Clinically speaking, C. pneumonia causes what we broadly call atypical pneumonia. It's often termed walking pneumonia, along with bronchitis and severe sinusitis. It spreads easily from person to person via simple respiratory droplets, just coughing or sneezing on someone.
Is it relatively common?
Extremely common. The text actually states that by early adulthood, the vast majority of people have measurable antibodies, meaning almost all of us have caught this specific bug at some point in our lives, probably just assumed it was a really bad lingering cold, eventually got over it.
But there is a really weird kind of dark connection mentioned in the text regarding this bug, a link to atherosclerosis.
Yes, the heart disease link. This is honestly one of the most intriguing and hotly debated frontiers in modern cardiology.
You're telling me a common respiratory bacteria might literally cause heart attacks?
There are several very strong lines of evidence suggesting it. Pathologists and researchers have actually found C. pneumonia DNA and whole bacterial proteins physically inside the hardened atherosclerotic plaques taken from human arteries.
So it's actively living deep inside the clogged artery.
It is physically present there. And the leading theory is that the chronic low-grade inflammation caused by the persistent bacteria in the artery wall actively contributes to the initial formation or the ultimate instability of that dangerous plaque.
That completely changes the game. We usually blame heart disease almost entirely on, cheeseburgers, bad genetics, and smoking.
We absolutely do. And to be perfectly clear, the source material explicitly notes that this remains a subject of intense ongoing study, and there are conflicting trial results. Massive clinical trials where they actively treated heart patients with long courses of antibiotics to prevent secondary heart attacks haven't been the absolute slam dunk we initially hoped for. But the association is incredibly real, and it fundamentally challenges our long-held assumption that heart disease is purely a lifestyle and metabolic issue. It very well might have an infectious spark.
That is just wild to think about. Okay, moving on to the third and final species. Section 5. Chlamydia psittaci.
Psittacosis.
Parrot fever.
Or ornithosis, depending on the bird. This is classic zoonosis.
The natural biological reservoir for this one is entirely birds.
Correct. Psittacine birds, your typical parrots, parakeets, macaws, cockatoos are the really classic carriers. But it widely affects poultry, too. Turkeys, ducks, even common city pigeons.
How do humans actually catch it? Do you have to get physically bitten by an angry parrot?
No bites required at all. It is primarily an airborne disease. But it's not strictly from a bird coughing on you. It's mostly from dust.
Dust? What kind of dust?
Dried bird excreta.
You mean dried bird poop dust?
I do indeed. When an infected bird creates waste, it falls into the cage or the coop and it slowly dries out. It turns to a very fine powder. When the owner or the farmer goes in to sweep or clean the cage, that highly infectious dust aerosolizes perfectly into the air, and you breathe it right in.
And unlike the other chlamydia species, this one doesn't just stay localized where it lands, right?
Correct. Trachomatis stays strictly in the eye or the genitals. Pneumonia stays fairly well contained in the lungs. Psittaci enters through the deep lungs, but it rapidly invades the bloodstream. It causes systemic sepsis.
So it goes fully systemic.
It goes absolutely everywhere. It travels rapidly to the liver and the spleen, causing them to acutely enlarge. Then, strangely, it settles back into the lungs, causing a very severe, patchy, aggressive pneumonia. Patients get splitting, excruciating headaches, dangerously high fevers, and severe muscle aches.
Is it a deadly infection?
The text gives a mortality rate of up to 20% in the pre-antibiotic era, or if it is currently left completely untreated.
1 in 5. That is an incredibly high mortality rate for a bacterial infection.
It is a profoundly serious pathogen. It's largely considered an occupational risk, now heavily affecting poultry processing plant workers, veterinarians, and pet shop owners. But it deeply serves as a reminder that chlamydia isn't just a purely human problem. It's a highly adapted family widely distributed across nature.
So we've got blinding eye disease, permanent sterility, potential heart disease connections, and a genuinely deadly bird fever. Why exactly is our highly evolved human body so incredibly bad at fighting this thing off? That brings us to Section 6, Immunology.
This is the core tragedy I alluded to much earlier. The text states very clearly, infected hosts do produce specific antibodies. We definitely make them. Our body absolutely sees the bug.
So we fire our bullets at it.
We fire the bullets. But the text immediately adds a very depressing caveat. These antibodies possess little to no protective effect.
Why is that? If we see it, why can't we kill it?
Because the bandit is already inside the house. Remember, chlamydia is an obligate intracellular pathogen. Antibodies are large proteins that float freely in the blood and the mucosal fluid outside the cells. They physically cannot walk through a solid human cell membrane to get to the bacteria hiding deep inside that protected vacuole.
So the bacteria is effectively completely invisible to our primary weapons once it's inside the safe house.
Right, so the infection persists indefinitely. Clinical relapse is incredibly common, but the real devastating problem isn't just that the immune system fails to kill it quickly, it's that the immune system essentially overreacts out of frustration.
Hypersensitivity.
Yes. The permanent tissue damage, the blinding corneal opacity and trachoma, the severe tubal sterility and PID is very often caused primarily by reinfection.
So catching it the second time is actually worse.
Much, much worse. The initial primary infection primes the host immune system. It creates huge numbers of hypervigilant memory T cells. When the host inevitably catches chlamydia again, those specific cells instantly recognize the antigen and they essentially go nuclear. They launch a truly massive, overwhelming inflammatory response.
And that sheer wall of inflammation destroys the surrounding healthy tissue.
It triggers the dense, rapid scarring. It is classic friendly fire. The immune system is trying so incredibly hard to kill the hidden, protected bacteria that it inadvertently destroys the host's own eyelid or fallopian tube in the violent process.
This perfectly explains why vaccines have been so incredibly hard to make for this.
It is the absolute primary scientific hurdle. The source specifically mentions that historically, early human immunization trials were an unmitigated disaster. By actively vaccinating people, researchers inadvertently primed their immune systems perfectly. When those newly vaccinated people naturally encountered the real bacteria later on, they actually developed vastly more severe, rapid-scarring disease than the completely unvaccinated control groups.
That is a literal nightmare for a medical researcher. You try to protect a population and you actually make them vastly more vulnerable to permanent damage.
Exactly. You gave them the dangerous hypersensitivity without actually giving them the protective immunity. Developing a safe vaccine that effectively kills the intracellular bug without triggering that catastrophic scarring response is the holy grail of chlamydia research, and honestly, we just aren't there yet.
So if we physically can't vaccinate against it, we have to rely entirely on diagnosing and treating it rapidly. Section 7. Diagnosis. How do we actually find this tiny stealth pathogen?
Diagnosis has evolved a massive amount over the decades. It used to be incredibly difficult and slow.
The text lists a few major historical and current methods: cytology, culture, serology, and nets. Let's break those down.
Cytology is essentially just staining, right? Just looking at a smear under a microscope.
Yes. We briefly mentioned the old iodine stain for C. trachomatis. It's cheap and it's fast, but it is deeply not very sensitive. You can also use a more complex Giemsa stain. This specific stain beautifully turns the tough elementary bodies a deep purple and the fragile reticulate bodies a lighter blue.
That actually sounds quite pretty to look at.
It gives you that really nice, clear visual of the biphasic life cycle in action. But again, you critically need a highly skilled microscopist staring through a lens to spot these exceptionally tiny cellular inclusions. It is incredibly easy to just miss them entirely.
Then there's physical culture, actually growing it in a lab. But as we established, you can't just streak this bug on a plate of warm agar jelly like E. coli.
No, absolutely not, because it's an obligate intracellular pathogen. You fundamentally need a layer of living, breathing host cells.
The text mentions utilizing McCoy cells for this. McCoy cells are a very specific, immortalized cell line originally derived from mouse fibroblasts. To successfully culture chlamydia, you have to take the patient's fluid sample, carefully put it on a delicate monolayer of these living McCoy cells, and then this is the truly crazy part. You very often have to put the whole plate into a centrifuge.
Wait, you actively spin the living culture plate?
You rapidly spin the plate in a centrifuge to physically forcefully drive the tiny elementary bodies down onto the surface of the cells to artificially encourage infection.
That sounds incredibly aggressive for a delicate lab procedure.
It is very aggressive. And then you usually treat the living host cells with a mild cellular poison called cyclohexamide.
Why on earth would you intentionally poison the very cells you're desperately trying to grow the bacteria inside of?
Because you actively want to stop the host cells from dividing and consuming nutrients. You want to artificially slow down the host's normal metabolism just enough so the invading chlamydia has vastly less competition for the available energy pool. You essentially handicap the host so the parasite can fully thrive.
That sounds incredibly labor-intensive and wildly complex. Costly and arduous is exactly what the text says.
It absolutely is. It takes days to yield a result. It strictly requires a high-level, sterile laboratory. It used to be the absolute gold standard for legal diagnosis, but it's just completely not practical for mass, rapid public screening.
So what exactly do we use in modern clinics now?
NATES, nucleic acid amplification tests.
Which is essentially just PCR, right?
Essentially, yes. We completely stop looking for the whole bug, and we just look for its specific DNA footprint. We chemically amplify very specific sequences of the chlamydia genome millions of times over. The text declares it the absolute test of choice for all genital infections today.
Why is it vastly better than culture?
Massive sensitivity and specificity. It can accurately find even ridiculously tiny amounts of fragmented bacterial DNA. And crucially, because it's so incredibly sensitive, you don't necessarily need a deep, highly uncomfortable physical scraping of mucosal cells anymore. You can just use a simple first-catch urine sample.
That is a massive game changer for getting people tested.
Huge. Men especially can just provide a quick urine sample in a cup. It's totally non-invasive. That single fact makes massive population-level screening programs actually feasible. You can accurately test thousands of asymptomatic people easily and cheaply. It has largely replaced the older, arduous methods almost entirely across the developed world.
Okay, so we've successfully found it with PCR. Now we urgently need to kill it. Section 8, treatment. We established earlier that standard penicillin is weirdly ineffective because of that bizarre cell wall paradox.
Right. Standard penicillin merely causes those defective, bloated forms, but simply doesn't fully clear the underlying infection. We absolutely need something else.
The text specifically lists azithromycin and doxycycline.
These are very different classes of drugs. They are protein synthesis inhibitors. They don't target the physical cell wall at all. They specifically target the bacterial ribosome.
And the ribosome is essentially the cellular protein factory.
Yes. Even though chlamydia steals all its energy from the host, it still strictly has to build its own unique bacterial proteins that survive and replicate. It has its own ribosomes. And importantly, bacterial ribosomes are structurally different enough from human ribosomes that we can target them safely without destroying our own cells.
So doxycycline effectively just shuts down the internal factory.
Exactly. It starves them of vital proteins. For standard genital infections, the clinical standard is usually a reliable course of oral doxycycline taken for exactly 7 days. Or alternatively, a single massive megadose of azithromycin.
That single-dose option seems like a massive advantage for broader public health compliance.
It is remarkably huge. We use a strategy called directly observed therapy. A clinician can literally watch the patient swallow the pill right there in the clinic. You know with absolute certainty they are fully treated before they leave. With a longer 7-day course, human nature kicks in. People forget a dose, or they stop taking them early the second their uncomfortable symptoms go away, and that leads directly to treatment failure and continued spread.
And what about for trachoma, the blinding eye disease in developing nations? You can't just hand out single pills and solve it, right?
No, the strategy there is vastly more comprehensive. It's the SAFE program, S-A-F-E. It's a massive, ongoing WHO global health initiative.
Break that acronym down for us.
S is for surgery. You absolutely have to physically operate on the eyelids that have already turned inward. You have to surgically correct the severe trichiasis to physically stop those eyelashes from continuously scratching the cornea. That is immediate vital damage control to save whatever vision is left.
Okay, that makes sense. What's the A?
A is for antibiotics.
Ooh.
But crucially, not just for the visibly infected individual. In severely hyperendemic villages, they utilize mass drug administration. They systematically give oral azithromycin to the entire community, healthy or not, to massively lower the total environmental reservoir of the bacteria.
Yeah.
F is for facial cleanliness, simply educating and equipping communities to regularly wash children's faces, physically removing that sticky, purulent discharge that actively attracts the specific fly vectors we talked about earlier. And the E.
E is for environmental improvement, digging deeply for better, clean water sources, building sanitary latrines to fundamentally reduce the massive local fly population.
It's an incredibly holistic approach. It's not just a medical pill, it's a massive societal infrastructure project.
It absolutely has to be. You physically cannot just treat the localized bacteria with a pill if the exact same dirty environment just keeps immediately reinfecting the local children the next day. You have to fundamentally change the entire ecosystem.
And finally, what do we do about the birds? For C. Cetacea control.
Control there is almost entirely about rigorous agricultural quarantine. Commercially imported birds, parrots, macaws are often legally mandated to be treated with prophylactic tetracycline laced directly into their feed for weeks before they are ever allowed to be released to commercial pet stores. You essentially have to clear the silent carrier state in the animal to firmly break the chain of transmission to humans. We have honestly covered an incredible amount of dense scientific ground today. From the deeply bizarre, energy-stealing molecular biology all the way to the massive global impact of blinding trachoma. As we begin to wrap up this specific deep dive, I'm trying to synthesize the overarching personality of this strange organism.
It certainly is a highly distinct one. If I absolutely had to personify it, I would say chlamydia is the ultimate biological stealth operator.
Stealth operator, I like that.
Think deeply about everything we discussed. It enters completely silently. It specifically hides deep inside your own host cells, actively preventing the cellular lysosome from ever destroying it. It efficiently steals your hard-earned energy so it literally doesn't have to carry its own heavy metabolic baggage.
No.
And very often it physically hurts you, not by violently attacking your tissues directly, but by slyly tricking your own powerful immune system into permanently hurting itself through massive scarring.
It literally uses our own primary bodily defenses directly against us.
It operates entirely on the evolutionary principle that silence equals survival. By remaining completely asymptomatic in so many millions of people, it absolutely ensures it gets seamlessly passed on to the next host. If it rapidly killed everyone it infected instantly, like, say, Ebola does, it would burn itself out very quickly. By staying deeply quiet, it successfully spreads to hundreds of millions.
It is genuinely chillingly effective when you look at the raw numbers.
It is, without question, one of the single most globally successful pathogens in all of human history.
Well, on that slightly unsettling, very clinical note, we have finally reached the end of our dive today. But right before we go, I have a final sort of philosophical thought for our listeners to mull over.
Let's hear it. I'm intrigued.
We generally tend to think of the concept of evolution as a constant march toward getting stronger or getting much more complex. We went from basic single cells to complex fish to monkeys to eventually humans. We built massive cities, we invented Wi-Fi, we place a massive cultural value on total independence and self-reliance. But chlamydia highly successfully went the exact opposite way.
It underwent intense reductive evolution. Yeah.
Exactly. It completely threw away its own genes. It intentionally threw away its fundamental ability to make its own energy. It completely threw away its own protective cell wall. It actively became far less complex in order to become far more globally successful. It made an evolutionary decision that total, utter biological dependency was a vastly superior survival strategy than trying to maintain independence.
That is a genuinely profound thought. In a human world where we so highly value utter autonomy, chlamydia perfectly proves that absolute, unyielding parasitism, total, complete reliance on another living thing can actually be an incredibly stable, highly successful evolutionary peak.
Maybe the smartest guy in the proverbial room isn't always the one doing all the heavy lifting and hard work. Maybe it's actually the one who silently figured out how to get absolutely everyone else to do the hard work for him.
A deeply disturbing but highly medically accurate lesson directly from the microbial world.
Thank you so much for joining us and listening to the deep dive today. Wash your hands, strictly utilize those screening programs. Maybe definitely don't inhale strange bird dust, and we will absolutely see you next time.
Stay intensely curious, everyone.